other? Okay. Or you think because it's going to...
So you need to use both mics, unfortunately. If that's okay with you guys. Yeah.
We're going to talk into this one.
Okay.
Okay, if that's there's no point talking to both phones. It's not going to get recorded. We're just going to use that.
Yeah, we'll just use that.
Oh, I see. Okay.
So just pass this along.
So you're going to talk to that and this at the same time?
Yeah. That makes no sense. Just use one.
Yeah, we're just talking to this.
No, he's saying this isn't connected to the... That's connected to the live stream. This is
God,
connected to the room.
Sorry, that's it. If Dr. King can-
Okay, just have one mic for all of us
Okay.
then. Yeah.
Oh, wait, I think I turned it on. I'm putting mine down. That's okay. I don't know.
Hello? Hi, everyone. Can we have your attention, please? Great. We're your TAs. My name is Yenna.
I'm James.
I'm Jessica. I'm Robin.
And we're going to take you through the next 2 assignments that are due for this course. Some important reminders. Your next assignment is due November 1st at 11:59 p.m. It's your visual summary assignment. And we're going to take you through some tips for you to prepare excellent visual summaries. November 4th to 8th, there's reading week. November 20th, the review paper draft is due if you'd like to send it to your TA for comments before submitting it for grading. The final review paper is due December 6th.
Okay, so we're just kind of going to go over a few tips that we've sort of, I guess, acquired over the past few years of marking, and just kind of highlight, I guess, important things to know for the visual summary. Here you go.
Oh, okay, that's me. Okay. So so the visual summary is a single-page panel, so depicting essentially the entire paper and the major points that they were trying to get across. So in terms of picking your articles, I saw that many of you guys already have picked one. Make sure it is a primary paper from any neuroscience-related journal. It should have been published in the last 5 years, and ideally it does not have a visual summary. We would just prefer that just so that you guys can maximize your creativity in your visual assignment. And kind of couple things that we caught in the past 2 years is that a lot of students forget to cite their articles in their summaries, so please make sure that is included in your visual summaries as well, and your name.
Okay, so just going over what a primary research article before we start picking our articles if we already haven't. So primary source is going to be something that was written and something the experiments were done by the authors themselves. So things to look for in a primary paper versus something like a review paper, a meta-analysis, or a case report is going to be methods and materials and results. So they're going to describe the methods that they did themselves and the results that they found as well. If you're not sure about any of the papers that you have, feel free to send it to us. Our emails are on this slide deck, and our emails are on Quirkus as well, so just reach out and ask us if you're not sure. And one of the things, a lot of the articles also will let you know at the top or somewhere on the side if they're a reviewed article or an original article. In the Nature papers here, they say review, they say article, they'll say original research at the top or something like that. So look around in the article, but again, just let us know if you're not sure. Cool.
Remember also, the article you're selecting for the visual summary is the same article you're going to be reviewing for your final paper. So make sure when you choose your article, it's a primary research article. And it has sufficient content for you to critically analyze as part of your final review paper. And when you pick an article, try to pick one that you actually feel interested in because you're going to have to read it a lot, and you're going to have to read papers related to that topic. So pick an article on a topic that interests you. When you prepare your visual summary, keep in mind you're trying to tell a story. Can I have your attention, please? Cool. You're trying to tell a story with your visual summary. The reason we emphasize this is because in the past we've received a lot of assignments that show a lot of different pictures and don't really tell us what the paper was about. Your visual summary, the main objective is to tell the reader, without reading what paper that was, what the main idea was. You need to tell us what was the objective of that paper, what did they measure, how did they measure it, what was their hypothesis that they were testing, how did they test that hypothesis, and what did they find, and why was that important? So it's like IMRAD, intro, methods, results, conclusions, discussion. You're doing that in pictures. And try to minimize how many words you use in your text or in your summary, because it's going to be harder to follow the more words you have. So use pictures to tell a story. And reference the grading rubric when you prepare your visual summary, because that's what we're going to be following along, and try to see what we're going to give you in each domain.
All right, so this is the example that was posted on Quirkus for you guys. I don't know if you guys had a chance to look at it yet. So this is one of the decent ones that was published in, or that was handed in to us past couple of years. So a couple of things to kind of point out why this was a good visual summary, and kind of the elements that you guys want to maybe take into account when you guys are creating your own. So there is an effective use of visuals over here. So there's a good balance between text and figures. So the figures are effective in terms of demonstrating the methods, as well as the text is a good complement to the figures to show what the authors did and the results are. We kind of discussed it ourselves. We would say that there was a little bit more words on this visual summary than that we would have preferred, but it's still not too bad. The methods and the results are very clear from this summary as well, so it's very clear to see that without even reading the paper, that we would, as markers, we'd be able to understand what the major point of the paper was. As well as another thing that a lot of students missed in the past couple of years is to define the acronyms. So it might be very obvious to you guys, but when we're kind of sifting through 50, it's kind of does get confusing. So just please make sure to define the acronyms that that the authors may be using. Kind of last two things, paper being cited, we've mentioned that before. What we found was really effective was when students included a kind of a summary statement, as you can see in the top right yellow box. And so this kind of concluding sentence to show kind of big picture what the authors are showing was important in terms of kind of showing what the authors concluded and kind of summing up the methods and the results that was shown through this visual summary as well.
Okay, so we agreed that this visual, you can go back. It was pretty decent, but ideally way more pictures, less text, because we want to be able to look at it and tell a story versus having to read it. Also, you don't need to include every detail of the results. We just want the main result, the main impact, like what's the overall summary. You're going to tell us the details in your report, but for now, we just want the big picture. So a lot of papers actually have their own sort of visual summaries on them, like cell papers and things like that. So don't use those articles, but you can kind of look at them as like an example. They're almost a little too simplified for the purpose here, but just an idea of like you want to be using pictures to tell a story again. And yeah, just with a nice summary statement at the end. Yeah, okay, what's next? Okay.
Before we move on to the literature review, I just want to say when you prepare your visual summary, keep in mind the key information that we want to see there. My supervisor always says PICOS. PICOS means participants, intervention, control, outcome, and study design. And you want to include those key elements in your visual summary, so at a glance we understand who your participants are. So that could be mice, but you should tell us what disease model the mice represents. Don't just tell me it's like C number something, 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 because we don't know what that is. So tell us it's mice, it's whatever C something mice, it represents Alzheimer's disease, and then there's a control. So that tells you who the participants are and what the study design was roughly. And then we want to hear what the interventions were. So what are they testing? What's the hypothesis? What are they doing to test their hypothesis, the intervention? So tell us what the intervention is and if there were any control. So what was the control? Did they use saline? Did they use something else? What interventions did they use? Next is controls. And then outcome. What outcomes did they assess? The title says anxiety, but you don't tell us like how the mice were assessed on aspects of anxiety. What kind of maze did they use? Were they like on a rod? If they fall off a rod faster, does that mean they're more anxious? Like, tell us what experimental paradigms were used to measure an outcome, and explain to us, contextualize what that outcome means to you. So don't just give us the name of some test they used. Tell us what that test was and what it measured and why it's important. And if increase in score or decrease in numbers means good or bad or something.
I guess we also we don't share the methods each of the basic papers.
Yeah, the idea is to tell a story, but you can't just tell a story with acronyms or anything. You need all the key elements, right? So PICOS, participants, interventions, controls, outcomes, and study design. Cool.
For some basic science papers, when we say methods, we don't mean like we want to see the qPCR data or a Western blot or something. We want to see like what that means. So just like, oh, the mouse had a higher level of this protein, and then show that in a visual. Don't show us like the physical picture of a microscope or something like that.
Okay, so that's your visual summary, and now we're going to move into the literature review. Are there any questions about the visual summary before we move on? Nope. Okay, if there's anything that comes up, feel free to email us or come up to us after class, okay? So the literature review assignments due later in November, and you have the chance to submit this beforehand to your TA, so you can get some feedback in before you do a final submission. So this assignment's going to be based on, it can be based on the same paper as your visual summary. If you feel the need to change it, 100% do it. You're going to take the paper you chose, and you're going to give us a bit of an overview about that subject, but you're also going to compare the paper that you did to other information in the literature to look at were the results consistent with other pieces of information, and was there any information that was missing to draw some more conclusions on it, and where can you go moving forward with this research. So for this assignment, it's going to be 1,500 to 2,000 words, and you're going to make it it's going to be about single page, 3 pages, give or take, and that's not going to include any figures used, references, and an abstract as well, okay? So those are all going to be additional, and we do encourage you to use figures from the research paper itself to include in your paper. Not mandatory, but generally helpful as well.
All right, so kind of looking at the outline of the literature review that you guys will be writing. At the very beginning of the review article, you should be writing an abstract on the paper. So it'll be about a 250-word summary outlining kind of the introductions, the procedures, the methods, the participants, the acronym that Yana just mentioned should be also included in there, the relevance of the results as well. So the next 3 are kind of fairly straightforward, so background and introduction. So talking about writing the informations that us as readers need to understand the results and why the authors decided to do this research would go into the background and the introductions. Keep in mind, just try not to just use the citations that the paper that you guys are reviewing used. Try to kind of go outside of it and see if there are other things that you guys want to include as well too. So major results are kind of going through the major findings and the figures that the article shows. You guys don't have to go through every single major results as well as the supplementary figures as well too. You guys would be just writing forever depending on the article that you're reviewing. So you guys just have to pick few of the major ones and just kind of review it and show what the authors are showing through those graphs or figures. Discussions would be talking about the impacts of the findings, comparing it to outside literature. If there has been literatures that have been published after that your paper has been published, definitely would be in your best interest to kind of refer to it and talk about how it adds to, or if it supports or does not support the article that you are reviewing as well. The last 2 parts, so critical analysis and future direction. I don't know if you guys will agree with me, but when I was marking the review papers in the past, that's where a lot of the students that put a lot of work into those sections, those were the students that did very well. So I would highly suggest putting a lot of focus into these, the critical analysis and the future direction. So critical analysis would be what kind of experiments would we need to perform, and that would be kind of in the context of the discussion that you wrote in the previous section. be talking about if there's any inconsistencies or gaps compared to the other sources that you reviewed. Future direction would be based on what the authors have concluded and what we know about the field now. How would you push that field forward, and what kind of research would you suggest that the authors could do next?
Okay, so basically, oh, sorry. We're not Britney Spears, sorry. I guess a good example of what we're kind of looking for, but more simplified. So it says literature review. Technically, literature review in like research lingo means a complete analysis of all the literature, and you didn't do any experiments, you just talk about experiments that have been done. That's not what we want. So it's kind of more, instead of like a literature review, it's more like a news and views. So some journals, like Nature, actually do this. If they have a really cool or high impact paper that's gonna happen like later in the journal, to kind of like hype readers up a bit, they'll publish a news and views, which basically is like a little summary about the paper they're about to look at. But first of all, as James was saying, like it outlines what are the gaps in the field, what are the major results, and then it kind of like critically analyzes them, or assesses them, and kind of, basically, it's like if papers are gonna maybe cause some conflict in the field, they're gonna address it here, and then a future directions section. So this model is like super simplified, but just if you guys want, you can always like look for these online and kind of get a general idea of the layout we're looking for. So for example, you're gonna have some sort of creative title, you're gonna have a complete abstract, so that's something that's different, and you're gonna have a proper citation for your journal, put your name, and put Dr. Ju's name after that, I like that. So you're gonna start with your introduction. At the end of the introduction, usually there's like, almost like a little summary of what the paper, like everything you're about to talk about in terms of like all the results and future directions right at the end of the intro. It's written more into your guidelines, so you can always message us if you need. But yeah, so you want to highlight the gaps in the field, and then you're going to move on to summarizing the critical findings in the results. So I don't know about you guys, but in the past, when I had students also cite external papers in the results section to kind of like support the results, be like, oh, the authors found this similar to like, or it supports this idea in the field. And then they cited another paper in the results that kind of, I guess, added a little more confidence to what they were saying. You don't need to add your visual summary, but you will have a few visuals in it, some key experiments that I guess kind of go with your text. Yeah, so then you're gonna, let's see, yeah. Okay, so then you'll get to the critical analysis and future directions, and again, just emphasizing what James said, this is the most important part of the paper. We really look for like creativity here. We want to, basically what we're looking for is, do you actually understand the paper enough that you can design a new experiment and you know what needs to be asked, I guess? Like what's remaining in the field to be asked and how you're gonna address it. So this part is really, I guess, how we can tell if you understood the paper. So that's why we kind of like emphasize, I guess, the marking for this section. You know, we don't need too many details of like an entire experiment, but we need to be able to see like, I guess a few examples of different things you could try and maybe like what your controls would be. Yeah, you can always like email us again if like you're getting a little confused.
Remember, the point of the final assignment is to prove to us how smart you are, and that will earn you an A. And the way you show us how smart you are is by showing us how well you know the paper you've chosen and other literature relevant to that topic. So not only do you need to be an expert in the paper you've selected, you need to show that you understand how important, or not important if your paper is shitty, how important that paper is to that field. Why was it important? What did they do that was new? How did they do it? Why do we care that they did this in a new way? Show us you understand the paper and the field. And that's what we mean by contextualizing the results.
Yeah. And I think there's even like a limit to the citation, so don't worry, you don't need to summarize the field again. We're just looking for like key points to be like, this is what's happening in the field, this is how the paper fits.
But don't list everything you know about the field. Show us that you can think critically.
Oh yeah, so again, if you do read any of these like news and views articles, take it more as like an example of an outline. Yours needs to be much more detailed than this, but yeah, that's the idea. Who's next?
Okay, so just another picture of the rubric that's up on Quarkus as well. Definitely take a peek at it before you start writing your review, before you finish your review and submit it to us. Some tips again, you can use the same articles you use for your visual summary. You can also change it if you want to. And we try not to. It makes it so much easier for you. It'll make everything that much more clear and you'll have a better understanding of the information as a whole. And really try to connect the information that you find within that paper and all the conclusions that you draw. Make those connections to other pieces in the literature and everything else that you've learned in this course and in other courses to try and really create that whole picture and the story that we're trying to see from this assignment. But yes, definitely look at this rubric before you start and finish this assignment.
Okay, so now we're going to try something. We're going to take our beautiful TA Yena's review, and we're gonna mark it all together. Isn't that nice? Okay, maybe Yena, you want to start off?
So the reason we chose mine is because we can rip on it as much as we want and not feel bad. So if you see something and you're suspicious that that was probably a bad idea, feel free to raise your hand and be like, that's bad, and we can talk about why it's bad. Okay, great. Should we start?
Yeah.
Okay, so the title is very long. Ideally, you should have a shorter title that tells us what it means, but it's not all that bad because we kind of understand what the paper was about. But I'm biased, so.
Yeah, there is, I like the little citation in the corner. It's nice, it looks like a real paper. So yeah, it definitely starts off talking about the introduction, or in the introduction, sort of summarizing the field. Oh, is this the abstract? Awkward. Okay. Here, yeah, you go.
This is an unstructured abstract. Some journals require your abstract to be structured, and they'll have different guidelines on what kind of subheadings you should use. I prefer structured abstracts now, but sometimes you might feel like an unstructured one is better for you. But the key aspects you want to include in your abstract always is background and rationale, the methods, the results, discussion, and significance or conclusions or something along those lines. And the background and rationale should tell us what gap in the field this study aimed to address. So why they did this study.
Yeah. Just looking at the abstract right off the bat, the first word, MHC1, is an abbreviation. And we just want to make sure that we're, again, defining all of our abbreviations, even in the abstract. So let us know what it is so we don't have to look it up. Great. 2014, yeah.
So the background should be about 2 or 3 paragraphs. It shouldn't be that long. Again, the bulk of your mark is going to be focused or based on how good your critical analysis and discussion, future directions, and significance sections are. So keep your background short and pithy.
So yeah, you kind of want to start your introduction like as basic as possible. Be like, this is what this protein is or does. And then you get into the research field, the gap, and then what this sort of like last area here needs to kind of be a summary of like what you're about to say. Be like, this paper found this, this, and this, and it contributes this way. Like in just a few sentences. A lot of papers do this already. Like cell at the end of every introduction of a cell paper, if you read the last paragraph, you'll see this if you need an example. But yeah, so you want to start simple and then get like more detailed into the field and then summarize.
Your background should start broad and then funnel down so it's more narrow. So start with the big idea and then narrow it down, and then at the very end, it should tell us, so why do we care about everything you just said? And the reason is because it's super relevant to the study that you're about to tell us. So start broad and then funnel down.
Research overview. Okay, so then you're going to go, after your background, you're going to go into a bit of a research overview, so looking more at what's been done in the field, what kind of stuff that you're looking at based on what the paper is, if there are any connections to other pieces in the literature that this paper was based on, and if there are any papers that base their work on this one itself. So make sure you look, we posted photos of those databases, you have PubMed, you have the U of T libraries, go through those databases and see the related topics just to see where this fits in with the rest of the research as well. And you're gonna start doing a critical analysis of how this research has been done. So look at the methods that they're doing and the other methods that have been done in other papers as well, and also start looking at the results that they've gotten to see how they compare.
Yeah, so you'll you don't want to be too detailed in like a critical analysis here, but you do want to reference like the field a lot. So right off the bat, I like this. She cited different papers a bunch of times, probably relating to like, oh, they found this, which supports this idea in the field, that sort of thing. So you're kind of like starting the critical analysis, like getting a taste of it, but that's more for the next section. Also, the visual here is good. You can clearly read the axis labels. Please do not send us a paper where you've like shrunk it in PowerPoint so small that I can't read the axis labels. If you can't, then I can't. So make sure it's very clear. And then with your figure caption, they can be pretty, I guess, like pretty simple. Do we normally cite underneath the yeah. So if you're taking yeah. So if you're taking the visual from the paper directly and it's not one that you've made, you need to always cite the paper underneath in a small line. But basically we should be able to just also look at the figure caption and kind of get a gist of what it is. Do not copy and paste the figure caption from the actual paper. That's too detailed. We don't care. And it's, oh yeah, plagiarism, true. Sorry. But yeah, okay.
In the results, you might be tempted to just vomit out everything that you found in the results section of your paper. We don't want to hear regurgitation of the different numbers and metrics they use and what decrease and what increase. Tell us why, like what it is, what it represents, and why we care. Not so detailed that it should belong in the discussion, but enough that we understand what you're talking about when you say rod, or how do you pronounce it? The maze speed decreased or something, like just enough for us to understand. Do you have a question? No, so the interpretation that takes place in the discussion contextualizes the findings in the broader field of that topic. Whereas in the results, you're just telling us enough about the method and the findings that they used for us to be able to understand what that actually means. So if they use the Morris water maze, and don't just say, these mice stayed in this maze or this water thing for like 5 seconds, and these mice stayed there for 10 seconds. Like, was the difference significant? Well, yeah, what does the speed assess? Like, is faster better, is slower better? Like that kind of sufficient detail for us to understand what these numbers mean.
Yeah, so we don't want it to be like too interpreted here. It's more just like summarize it in your own words, and then you're going to analyze it in the next section. This is more just, yeah, relating to the field. Oh yeah, I think there's 2 pictures. So you should have 2 sort of images in your report. Again, just the important ones. We don't want to see all the data. Next. Yeah. Is there 3?
So I read this afternoon, this section under astrocyte activation. At the end of the paragraph, I was left asking, so what? Like, I just mentioned that in these different experimental paradigms, something increased and something decreased. And then I was wondering, so, well, what does it mean if it increased and what does it mean if it decreased? Why do we care about astrocytes and activated astrocytes? And how does this help us understand stroke, inflammation, and neuronal injury? And what do astrocytes do? Right, so at the end of every paragraph, you should tell us, so what? Important note here, when I did the assignment, we didn't have a separate section for critical analysis, so you're gonna see here that critical analysis being sprinkled throughout the results, but for your purposes, you should have a separate section titled critical analysis. In the second column on the right side, I highlighted an example that I thought answers so what better. And that is because I mentioned that the hippocampal slice cultures are different in the different paradigms, and then I commented that the slices do not contain peripheral immune cells, and therefore when they found that the different experimental paradigms exacerbated cellular damage, that meant that peripheral immune cells weren't relevant to the exacerbated damage that they observed. Does that make sense? So not only was I telling you that the different cell cultures were different across the experimental paradigms, I was telling you that in these cell cultures, they did not have peripheral immune cells, which means that peripheral immune cells were not relevant to the observations at the end. Does that make sense?
So this would, I guess, would kind of be like the critical analysis section, so here, Jenna is very bold and is proposing a mechanism for the field, right? But you can totally do that as long as you're supporting, like have a lot of supporting literature. Basically, we just want to say that you can think critically and be like, this is good, but also we want you to point out like, this, and this should have been done and wasn't. Or they interpreted this way, but it should actually be interpreted this way. Because scientists love to like overhype their own research and be like, oh, look, we just like cured cancer. It's like, no, you found a protein associated with it, but let's be clear. So that's what you kind of want to do. You're just like calling out scientists here. And like kind of like relating it to the rest of the field and being like, this fits here and here, but it doesn't fit with this. So a lot of the times you'll have like 3 papers that argue completely opposite things. That's okay. You could talk about it here and maybe why that would be or why they found the results, but other people found something different. So that would be also something important to address here.
To follow her cancer protein example, let's say they found that this protein improved cancer survival. You also want to demonstrate that you understand what this protein does, how they blocked it, and why it's important, and what it means in that protein signaling cascade, and how that signaling cascade is relevant to cancer. So you want to demonstrate that you have like a mechanistic understanding of everything that just happened in your paper. And this is your opportunity to show us how smart you are by taking us through the pathway and why it's relevant to that disease model.
Not the whole pathway.
Not the whole pathway, but only those parts that are central to your paper.
And besides critiquing results and the findings of the paper, also look at critiquing the methods. Are there different ways that this experiment could have been done, and would they maybe have produced different results? Would they maybe have been better or worse? And could you have found different things doing different ways? So look at results, look at methods, and just try and think critically about the ways that you could do this paper differently and what you can do to build on it as well.
Kind of to give a different take. So you don't necessarily have to fully trash the paper that you are going to be reviewing. So remember that this is a critical analysis, and part of that is also talking about what the authors did well as well. So talk about what methods were very good, what kind of if their logic was there, if their the methods that they used was kind of the gold standard, for example. So you can talk about the negatives, but also feel free to, and I definitely encourage you guys to talk about what the authors did well as well.
So let's do an example so you guys understand or get your brains warmed up in like how to critically analyze a paper. I recently read a paper that found that babies that were born preterm were less likely to have lifelong romantic partners. And this paper was published in like JAMA something, or like, I don't know, it was ridiculous. I thought it was a joke. So the study authors had a good rationale for why they selected that and why they thought preterm birth or low-weight babies might have long-term social deficits. But there are other aspects that may be relevant to long-term social relationships that are not fully explained by your birth weight. So these are some of the things that you may want to explain. So you might talk about why birth weight might be relevant to long-term social outcomes. For example, you might want to talk about preterm birth and inflammation and how that can affect neurodevelopment, and why that might have long-term repercussions for this baby's brain, and so on and so forth. But you also want to talk about other things that might be relevant to the thing that they were trying to measure. For example, maybe there's a confounding factor. So maybe the babies that were born preterm were more likely to have, I don't know, mothers that smoked and were cocaine abusers, and therefore they had a lack of role models in their lives, and perhaps then they were less likely to have long-term social relationships. Like, there's a lot of different things you could think about that can explain their finding, right? So these are the kinds of things you want to think about. Just kind of just take a step back and kind of question, like, what are they doing, and does this actually make sense? And cite different papers.
Okay, so for future directions, so you just did critical analysis. So again, like, it's differently worded when we did it. You want to have like intro, results, critical analysis, future directions, like very clear. But for future directions, I guess the kind of point is you're gonna almost like you did in your intro where you pointed out the gaps in the field, you're gonna come back here and basically be like, did they fill the gaps? So did they fill the gap? And if they didn't, what do they need to do to like fill it? What kind of experiment can you think of that might answer it? Or how could they have differently designed their experiment that might have better sort of answered the question they were trying to get? Basically, you want to either propose experiments that, well, a few maybe that they could have done alternatively, but you want to go beyond that and be like, okay, what's next for the rest of the field to do? If a scientist is reading this paper and is like, oh, I want to explore this more, what would be like the first experiment they do? So that's kind of what you're going to talk about and design briefly. So here she mentions like the control knockout mice versus, the knockout mice versus the control. Like, that's important for when we're reading your reports, if we can't sort of, I guess, like put the experiment you're proposing in our head, like put it together in our head, then we can't, I guess, totally market the most fairly. So we just like make it clear, but make it concise.
When you talk about future directions and you're proposing different ways you might think about X, Y, Z, you want to cite different papers that are already out there. It's great to have original ideas. They're very valuable, but the point here, you want to show us that you understand the literature. And citing different papers that support your idea helps you do that.
Like if you're proposing an experiment where you're like, no, they actually have to look at this water maze test instead. And it's like, oh, because this paper in 2012 showed that it's a better standard for measuring this type of anxiety. Something like that. So that's where you kind of like go and pull into the literature, just to like support your ideas. If you're proposing something, you want to like, I guess, sort of show us that we can believe you that this is a good experiment because you cited all these papers that support your idea. Oh yeah, and then you're gonna have your citation section. I believe there's a cap, right? I think last year. Yeah, 50, 20. So that's why we don't need like 100, but like we just want these sort of citations scattered throughout, wherever it's gonna help support your arguments, basically. And as long as it's the same format for citations throughout, then we're good. Yeah.
Try to avoid citing too many review papers. We want to see that we looked at the original papers as well and the original research done in the field, just so that we know that you guys have looked at the other research that's out there and how it's justified as well. Are there any questions? Yeah, we mentioned somewhere in the past, and I think it might be on the rubric as well, you can use any citation system that you feel works best for you. Just make sure to keep it consistent throughout both your visual summary and the literature review. Okay. Any other questions? Yeah. Yeah, people have been using BioRender, so you can try that one. I think it's the website.
So we understand you're all not artists. That's fine. Okay. PowerPoint, awesome tool. You use it well into grad school. So you can use PowerPoint, make some visuals. If you're taking clips, art, or something off the internet, you need to cite it. But yeah, PowerPoint's fine. You don't have to be too fancy. Some people are really good and like go full out. That's fine too. It's up to you. Do you have a question? Anybody else have a question? Where? Okay, yeah. Oh. Yeah. We'll get that Dr. Dude to post it for sure. Mm-hmm. Cool. We're excited, guys. Also, like, I know we talked a lot today, but we don't bite. Like, you can also send us like your paper if you're not sure. I remember last year I went over a few students' papers to be like maybe try, like they'll send 3 and be like, which one do you think would be best? I want this one. And be like, okay, yeah, I support you. I think that one's a good one. So like you can always ask us if you really need. We can also like help you throughout if you're kind of want to propose experiment but not sure if it goes in critical analysis or if it goes in future directions. Yeah, you can always come talk to us. Okay, any last questions? No. Oh, yeah. Yeah, so the rubric, but also Dr. Jupe has a posted sort of like outline. So it'll tell you in the outline on the PDF online exactly like what you should say in each section. And then the rubric really helps with that. Like we really rely on the rubric when we're marking. Yeah. Oh, so for the results figures, you don't need to create your own. I mean, you're welcome to if you like, but you can use like the main, or I guess what you think are the most important results from the original paper, and then just cite that. Yeah, you don't have to come up with your own. You already did that with the visual summary. Okay, y'all want a 10-minute break or something? I think Dr. Ju's gonna show up in 10 minutes, so yeah. Okay, 10 minutes.
So just a reminder, actually back here. So some quick resources before we wrap up. If you ever have any questions, please email us. Our emails are on the slide and on the website as well on your syllabus. The HMB office is in Wetmore if you have any questions as well. And also, before you send this out, if you have any questions about your writing, the college writing centers are really helpful and can help you review your writing. They'll give you feedback, but we're also here to do that if you need anything. The writing website also has some tips on writing literature reviews, which is super helpful. Just ask questions where you need to. And just a reminder of your upcoming dates. So if you want to submit your review paper before we grade it, you can send it to us as many times as you want, and we'll give you as much feedback as we can. So do that, and then the final due date is going to be December 6th. Okay? Cool. Send us an email, come up to us in person if you have any questions, and take your 10-minute break. Thanks, guys.
Okay, I think that was pretty decent. Good job. Oh. Thank you.
Nice. Do you ever just email your whole section at once?
No.
Okay, yeah. Well, I was gonna say, like, maybe I'll, like, go sit at New College for a bit, and if you have anything, I'll just sit there.
So
We'll do office hours together.
We can. So for the
Sorry.
report, we should say that maybe. For the report, we'll actually do like 2 office hours.
Okay.
Sorry again for all the hassle.
That's so okay, not a problem. I was more worried about, like, falling. It was just embarrassed or what, but it was fine. No big. All good.
Are you talking about the report?
introduce something.
Yeah, because I haven't read this yet, so I don't know. Or you should, like instead of doing this, you do more of this. Right? Yeah. Okay, sorry. You're saying what mechanism? You're saying this or this? No. Right. Yeah. You should leave it how it is. I like it. Are you saying you want to
Hey.
take it out?
Yeah.
You want to? Okay.
I'm 80% sure that he should be coming by after this to make a quiz lecture. Cool. Yeah. You're heading out?
Oh, yeah. Yeah, that's perfect. Either there or at the bottom.
Yeah.
So are all the images?
Yeah, I just been saying yeah.
Oh, okay.
There is a lecture.
And you don't need to do that. Just make sure.
Hey, what's up?
Okay, so I know you said you like really understand all the methods.
Yeah.
So in the studies that I'm planning on using, they use like a knockout mouse that they
Okay.
create,
Yeah.
and I don't really understand like how they
Create
the
them.
knockout
You
mouse.
don't need to understand how they do them. That's super complex. Way
Yeah,
beyond.
it was like-
Yeah, boxing is definitely not for me. No, just say that this is the knockout mouse they use and say what kind of control they would use as well. So just being like, this is the experimental mouse that they're using and why. And this is the control, how we're looking at healthy mice as well. Don't need to go into all that during it.
Okay, and so you don't have to go into the details of like how they do like protein like that. So
No, don't worry about that.
okay,
Not
thank
at
you.
all. Don't worry. Please don't. Please just tell us when it's a box race. I don't want to talk about it either. I just don't want to.
Like, yeah, they came up to show their thing right now. Yeah.
Are you done in the lab today or do you have to go back?
Because the student emailed me and she wants me to stay after class.
Sorry? That's fine. Good. Tuesday. No, my class is going to the bar after a test. So I'm going there. Really shouldn't. Oh, you're
If you guys have any, like, if you have your visual summary done and you do have quick questions and want us to scan something, you can just come up and we'll talk to you. But yeah, keep chilling. Dr. Joe will be here shortly.
Stay chill. Yeah.
I think like brainstorming thing. We'll give them a paper and tell them to like say things that are
Positive or negative?
like
Always ask
Pretty
say
cool.
if they have any questions about grants.
what I do.
Any questions about?
Wait. I don't know about a
That
wait.
sounds
Haunts you from the future.
This is Halloween, but forever. Cool, 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 cool.
Struggling with like defining.
Ready for my nap? Me. Me too. Who's your TA? Me? Robin? Plus? Oh. Whatever. Here.
And then this is like the person that happens in our mind.
Oh. Did you scoot to class?
Yes.
That's super
How do things go?
Good.
Thank you. Did you have time to kind of walk around and chat with students about their assignments? Did you have time to chat with students about their assignments?
We told them to come down right now, but there are a few people that came down. There was like, yeah, one guy like
We didn't think to walk up. No, we can walk up and around.
Okay, awesome.
We can like do a lap. We can do a lap. Okay, I'll start.
Yikes. Yikes. Team not here. Okay, cool. Right. There it is. All right. Okay, folks, if you don't mind, we're going to get going. Apologies, I'm missing the first hour, but I'm hoping that we can finish a little bit early. So you don't have to worry about no tonic at all today. If you are on no tonic, they will have like a little warning that you can actually add some comments and feedback. Please do so if you are, but if you're waiting for the poll, I decided not to have one today, mainly because I wasn't sure how many students would show up in the second hour. So what we're eventually going to go to is to talk a little bit about this concept, which is a healthy mind, a healthy body. And so what we're aiming for is to build the best possible healthy brain in this course over the next little while. That means that we have to talk about exercise. And the controversies around exercise, the fact that muscle is now known as a source for BDNF is like a really, really cool concept. It used to be that muscle was not directly producing BDNF, but we now know muscle produces BDNF. And we also know that sleep actually resets a lot of the functions that we need for the immune system, and we'll be talking about those factors as well. So in the last lecture, we talked a little bit about the fact that we don't really have good targets for depression, and we talked about the fact that even though we're living in the era of personalized medicine, which means that all of you have grown up in the era where genomics dominates, where we associate different genes or single nucleotide polymorphisms, we associate them with different disease states, or we think that there's a gene and an environmental effect. In 2019, we still don't have like a really good sense that there's a gene that's involved in depression, and that becomes a little bit of an issue. And we talked about that in the last lecture as well. And so the reason why we're really interested in this particular paper was that it hit all of the highlights that we were looking at. So we looked at the fact that neurotransmission might be affected, and we looked at the fact that different types of neurotransmitters, not glutamate's actually not that affected in depression. GABA is not all that affected in depression, but we know different types of systems like noradrenaline, serotonin, these systems are affected. And so this one paper that came out in 2003 actually linked a lot of those different pieces together. And it also implicated the fact that it's not just genetics. You can be a carrier or have the short form of the allele for the 5-HTT-PLR, but it doesn't mean that you will develop depression. It means that you might develop it, you're susceptible to developing it, but you need a stressor in your life in order for that to work. And so this fit all of the different pieces that we were looking for in our genetic model of things like depression. And in the last lecture, we talked a little bit about the fact that when we look at this shorter allele, that in fact the short allele results because there is a small missing piece of the 5' VNTR region, so it's deleted, and the actual gene itself for the serotonin transporter is actually not affected. So there's a problem with the promoter system, which means that we don't make as much of the mRNA. So we don't get efficient transcription of this particular transporter. And we went through some of the rationale and we went through some of the ideas that were associated with having a small deletion in this 5' VNTR region and why it led to this consequence of actually developing fewer of these mRNA transcripts associated with the transporter itself. And this is sort of where we left off. And it was a really exciting time. There was a gene, and that gene itself doesn't mean that you'll develop depression, but it means that you are at increased risk of developing depression if you have the right type of stress sometime in your life. And when this group out of New Zealand studied these individuals longitudinally, prospectively, so they started enrolling these individuals at a young age, then they followed them into adolescence, early adulthood, so it's a long study, about 847 individuals. They realized that if you had this allele, the S version, the shorter allele, with a deletion in the 5' VNTR, that if you encountered stress later in your life, that you might develop depression. You're at an increased risk of developing depression. The problem is, as we've been looking at throughout most of this course, if you look across all the literature, and you might be wondering, why are we doing a literature review? Why are you asking us to kind of compare different papers? And why are you asking us to look at other papers beyond the one that I've chosen to look at? And the problem is this. So that was a 2003 paper. And one of the things that we've emphasized over and over again throughout this course is this paper hasn't been replicated, or these findings have not been replicated in humans. And the problem is in 2009, another group came out and did a very different type of study. And this group, 6 years later, in a very highly ranked journal, the Journal of the American Medical Association, or JAMA, showed that there was no correlation, that it didn't matter, that if you had the short allele or the long allele, that there was no difference in the rate of depression. So that idea that the short allele is equal to depression if you carry it and then you have stress, this group found the exact opposite, that there's no correlation at all. So where does that leave us? Where does that leave us in terms of our understanding? So if you look, this is a meta-analysis, so analysis of published papers in journals where they are now looking at not 847 individuals, they're looking at 14,000 individuals. So do you trust data from 847 individuals or 14,000? And the logical answer would be, well, this obviously looks at more people, it will wash out any biases, and so this result must be the correct one. And there was a lot of debate between Caspi's group, the original 2003 paper, and this group kind of going back and forth. Like, why did you not find a correlation between the S allele and developing depression? And there were discussions about you're not examining depression, you're not looking at correlations of stressors. And so there was a lot of controversy around this back in 2009 that this paper was trying to also address. To make matters even more complicated, and if you're interested, this is the paper that you can go back and follow. If you're interested, a slightly more recent publication in 2011, also in JAMA, but specifically in the Journal of the American Medical Association in Psychiatry, did another meta-analysis. And here, I'm going to take you through what a meta-analysis looks like. So a meta-analysis is kind of like a review, and Yenna is actually an expert in meta-analyses. Yes, I'm talking about you. Sorry about that. So here, we have all of the published data, and these are all of the different papers, and all the papers are outlined for you here on the left. You can go back and look at each of these different papers, and then they want to know, compared to the S allele or the L allele, which one is more likely, based on the published information of many, many thousands of individuals, in fact, a larger study than the one that I showed you in 2009, their data is actually included in this subsequent meta-analysis. Does the data support that you have greater sensitivity to developing depression if you have the S allele, which is Caspi's original hypothesis, or is there no difference if you have the S versus the L allele according to the previous 2009 paper? So we're looking at the yet another meta-analysis to try to address this effect. So what this says, looking at many, many more thousands of individuals, and these are all the papers that they drew their information from, and what you should be looking at line here, this line says that if there's no difference, and this is the 2009 paper, if there's no difference between the S and the L allele, that in fact, all the data should line up right here in the middle at 0.5. It's like an equal probability, 50-50, right? So there's no difference between S or L. It's 50-50. It has nothing to do with this at all. And this is the 2009 paper. All of the data from all of the papers, if this is correct, should line up right here in the middle at 0.5. And if there is greater sensitivity to developing depression, then what we should see is that the data should fall to this side, toward the greater sensitivity to the S allele. You will develop depression. And if you are more likely to develop depression if you have the L allele, the long version, then most of the data should fall out here. So these are all of the different papers. These little dots that you see show you what they found, what they published. Was there greater S sensitivity to develop depression or not? And you'll see that some papers actually show that there's no difference. And the sizes of these boxes that you see here, so the larger boxes that you see down here at the bottom, sitting right at 0.5 where the pointer is at, and this is that 2009 paper, the Anya et al. Paper at the very bottom. If you look at all of the data, there are some papers that actually say there's no difference. S allele, L allele, you're at equal probability of developing depression. You're sitting right there in the middle at 0.5. Are we all good on that? All good? And this meta-analysis trying to make sense out of all of the published data that exists out there. Ultimately, what this last meta-analysis in 2011 showed by comparing all of the published data, including the data from 2009, the data that you see here, that if you look across all the literature, and you're not cherry-picking the literature to bias yourself, that in fact, the data shows very, very consistently that there is actually a strong trend, which is what you were seeing with this little arrowhead in red on the left, that all of the data actually shows that we have very strong evidence, at least in this meta-analysis, that the S allele confers susceptibility to developing depression. So you take all the data here, all these little dots that you see here running along the left, you pile them all up, that becomes like a lot of proof from published papers that the S allele actually is going to have a big effect on developing depression. Are you okay with that? Do you all follow that line of reasoning? Yes? What is the box size? Okay, great question. So what is the differences in the sizes of the box? Okay? So why are some of these boxes bigger? Why are some of these boxes smaller? And the size of the box represents the number of individuals that were used in the analysis. More individuals, bigger box. 14,000 is bigger than 1,000. Any other questions? Great question, by the way. Thank you for asking that. Any other questions? Yes? Yep. Yep. Correct. So it's a great question that your colleague is asking. So the last 3 studies that are at the very bottom of this particular meta-analysis, so an analysis of all of the other papers that have been published, they all show that they're sitting at 0.5. There's no difference for these 3 studies. So shouldn't they be weighted more? But the way that this meta-analysis is carried out is that it's looking across all of the data that's been published. And again, if you continue to look in the literature, this is 2011, we're sitting 8 years out, there are further meta-analyses, and the story is going back and forth. There is no clear-cut, easy answer for this. So I also had mentioned to some of your colleagues earlier, so some of you have been coming in to view your tests. There will be more test viewing times next week as well, and then the following week. I strongly encourage you to come in if you have the time. If you don't, I'm happy to arrange a time, and I've done so for a number of students. One of the things that is really interesting about this that I wanted to highlight for you, in each of the lectures that are coming up, the students have been indicating, oh, you know, I wish you would change something in lecture because it was not clear that this would be on the test. Or I wish you would change something in lecture so that I knew a little bit more of what to study. So just to give you a little bit of a heads up, there will be 2 multiple choice test questions from this lecture, this section that I'm presenting to you now. And this will be sort of one of the multiple choice test questions where you will be given some data like this and then just be asked to interpret the data. Are you guys going to be okay doing that? So can you tell me if it's something is statistically favorable to one allele versus the other? Are you going to be good with that? Okay, awesome. So I'm hoping that it's going to be good. You know in advance that you should know like what the sizes of these boxes represent. What do all these names over here on the left mean? Why is this line going down 0.5? And if you can understand and follow all of that, you'll be fine for that multiple choice. So here's where we're at. We're at a point where we're going back exactly where we were with Parkinson's disease. And the analogy, the parallels are actually pretty striking. So outside of the very small number of mutations where we have more alpha-synuclein, so this is genetic, and outside of mutations in DJ1 or pink, we don't have very strong evidence for genetic cause for Parkinson's, right? Do you remember that? You don't. That was term test 1. We don't need to remember. We forgot it all. Well, I hope you kind of remember that parallel. Most of it was unknown, of unknown origin. And so we're right back to where we began with Parkinson's. Parkinson's, we don't have a good model, and so we have to induce Parkinson's by injecting in a toxicant. Do you remember that? Yes. Okay, awesome. Thank you. Awesome. So if we take that same parallel, what we're doing here now, we don't have a genetic model for depression because there's so much controversy. We can't introduce the short form of the serotonin transporter because half the groups will agree, half the groups will not agree. And so we need to have an inducible model of depression. And this inducible model of depression, there are many, many different types, and I'm going to introduce you to some of these different types over the next little while, but most of these animal models involve inducing depression. So how do you make an animal depressed outside of sending them here to U of T? So there are lots of ways to do this involving stress. And one of these ways to induce depression is to generate a model which is known as CUS, or chronic unpredictable stress. And this chronic unpredictable form of stress means that at any given point of time, there is a stressor on the animal that they're not enjoying. So things like turning on a light when they should be in the dark, that's a stressor for the animal. They don't know and they don't expect it. It comes out of nowhere. So think about like what that means for human life. You're not expecting something bad to happen, then it happens, and that's a stressor on you. Or squirting water on them, also like uncomfortable for the animal, not expected, and it stresses them out. Their cortisol levels rise, and we've looked at cortisol levels rising. One large injection of cortisol sometimes induces depression in animal models. And so by doing this over a prolonged period of time, between 3 weeks and 1 month, makes these animals depressed. And these animals become depressed so that they're no longer interacting with other animals, so they tend to be not very sociable, and they will have some other phenotypes that are associated with them as well. So just before we get into other parts of this lecture, I also wanted to go back. Do you remember, well, it's like a walk down memory lane. Do you remember diet-induced obesity? Yes, okay. Do you remember that there was diet-resistant animals? So obesity resistance. So it turns out that there are some animals, even though they're technically genetically identical, if we do the same sort of chronic unpredictable stress, no matter what we do to them, they actually don't get depressed. We can't see signs of depression in these animals. And these animals are what we call resilient. They are less likely or unable to develop the signs and symptoms and the phenotype of depression. So again, it's very interesting. Studies have been actually done where you take resilient animals where you can't induce depression and you breed them together and they will also develop resilient offspring. So is it genetic? Well, the breeding suggests that it could be genetic. There's some kind of genetic factor that is being passed along. And you might also anecdotally, so it's not a great form of proof, but you probably know individuals that no matter what happens to them, they're like always going to be okay. Like it doesn't matter. Like they can have, their car could be like broken into, or they could have like a flood in their home, or they could have lost their job, or something worse could have happened to them all on the same day, and they're just going to be okay with it. They're going to be very, very resilient, and you probably know those types of individuals. And so it becomes a very interesting sort of discussion point. So we don't have a reliable transgenic model. We'd like one. We always want one in biology. If we can manipulate a gene and we know the signaling pathways, that's useful for us. And so we are always looking for these types of gene pathways, but we don't have one. 5-HTTPLR is not going to work. It's so controversial. So that original 2003 gene, that depression gene, isn't going to be a good animal model. So we use these induced models. And there are a number of ways that we can go back and check. And so this is the ways that we check for depression. So just keeping that clear. If this comes up on a short answer question, I want to be very clear on this. If I want to know if an animal is depressed, I will let them undergo a forced swim test, which I'll show you in the next video. There are different ways to do this forced swim test, and these are all different types or subtypes of a forced swim test. We can do a tail suspension test in mice, and the tail suspension test over the course of 2.5 minutes, you don't keep them there indefinitely, but over the course of 2.5 minutes, a standardized test, will this mouse continue struggling or will it stop struggling? Which one do you think is depressed? The one that struggles or the one that stops struggling? Stop struggling, right? So motor effects, etc. So using this tail suspension test is a way to look for signs of depression in an induced animal model after like chronic unpredictable stress. Are we good so far? Any questions? Is there anything I can clear up for you or any clarification questions? No? Okay, and in the next lecture, I'm going to come back and talk about some other models to induce depression, including foot shock models, forced separation models. These are different models to induce depression. These should not all be grouped together. I will make sure that I post that announcement for everyone or make it clear on the modules page. So these are the different ways here on the right that we can test after an animal has undergone chronic unpredictable stress, or after it's undergone random foot shocks, or after it's undergone forced separation. If it's depressed, we can do this tail suspension test, or we can do this forced swim test. And some of this looks like the following. And let's see if I can get this to work. Nope. So this is an animal that is, has not shown any signs of depression. So it's trying to do this climbing, it's swimming, it's being very how we identify these lab animals. And here you can see an animal that is depressed. So it's just floating. So it's not actively moving. So this is the forced swim test, one variation of this forced swim test. So again, the animal is active, it's swimming, etc. And so this animal not being depressed. Okay, so hopefully you have an idea of how this all works. And again, I'll come back and talk about this in more detail in the next lecture. And one of the things that I wanted to indicate to you is that we do have multiple models of inducing depression, and we have multiple ways to examine if the depression model worked. So I want to make sure at this point, is that clarification, is it good for everyone? Do you all understand? So can anyone tell me, like, of the 3 things that we've learned so far, what are the models that were on the previous slide that induce depression? What could we do? So chronic, unpredictable stress. There's another one, yes? Sorry? Learned helplessness is one that I'm going to point out. Anything else? So random foot shock, so that will induce depression. And then separation models. And so what we're looking at here in the middle are some more of these examples that we have been looking at. So if we have this learned helplessness model or learned hopelessness model, one of the things that we can do is at random intervals, just give a foot shock. And what do you think the animal will do if it's moving around and randomly gets a foot shock? What do you think the animal will do? It'll stop moving. And so when we see that it stopped moving, and we just randomly give it a foot shock, it doesn't know when it's coming, it just kind of gives up, right? So again, it's sort of a model that we use for learned helplessness. We can also call it sort of random foot shock models. Either one is good. And it's another example of an inducible depression model. We can also do forced separation or social isolation. So animals are very social creatures, and by isolating them, we can also induce depression, which we could then follow using like a forced swim test or a tail suspension test. They will show all the signs of like immobility, not moving around very much as well. And there's a lot of controversy as to whether or not some of these classically identified depression models are actually not better models for anxiety. So they look very similar in animals, and it's really hard to tell apart like anxiety and depression in animal models. And so some of the things like social defeat, is it really inducing depression? Some people say yes. So you put your mouse that you're interested in with a much bigger animal, and the bigger animal kind of acts like a bully, and it causes depression in the smaller animal. And this social defeat, is it depression because we don't see the animal moving, or is it a model for anxiety? And the data and the literature is a little bit sketchy on that. The other thing that we can do is also do direct intracranial stimulation of certain areas in the brain, and this leads to understanding that some areas in the brain are actually more vulnerable to depression, or are more important in depression than we sort of thought. So for example, especially in models for learned helplessness, these random foot shock models that you are seeing here, one of the things that we see is that when we look at the periaqueductal gray, which is what the PAG stands for here at the bottom in the blue arrow, in the periaqueductal gray and in areas of the brain like the nucleus accumbens, one of the things that we see is as the animal becomes more and more toward the right and they become more and more resilient, we actually see an increase in delta phos-B expression. And I'm going to talk about delta phos-B in a moment. And so this area of the brain, whether it's periaqueductal gray down here on the brain here in the middle, or we're looking at similar areas that it leads to, like the nucleus accumbens, the expression of different types of genes like delta phos-B, as well as action potential firing rates becomes important. So from this diagram, the thing that I'd like you to take away is really the delta phos-B element. So this year we're not going to be concentrating on the ventral tegmental area, action potential generation and firing, but it's really the top that I'd like you to concentrate on. And the rest of the lecture slides, the remaining ones, will be dealing with that. So Anything that is unclear for you at this point that I can clear up for you, spelling or anything else? Yes? So we're going to come back to that a little bit later, so if you can just hold on to that. Those are really tough to separate. So for a lot of individuals, and even clinically, making the diagnosis of depression and anxiety is like comorbid, like 3/4 of the time or maybe higher. It's really challenging. So we'll talk about depression and anxiety model. Make them a little bit more for you when we get there. Any other questions that I can answer for you? Yes? It increases in other areas in addition to the periaqueductal gray. I had mentioned PAG on that previous diagram, and the spelling for that periaqueductal gray, or PAG, is on this particular slide, but it is a transcriptional factor in a number of other areas as well. So we're going to forget about the action potential firing, and one of the things that we can also look at, in particular, in a circuit of the brain that we're very, very interested in, and this circuit is a dopaminergic circuit, not just the periaqueductal gray, but in the nucleus accumbens circuit, which is intimately tied to dopamine levels. It's intimately tied to motivation and reward and movement, all the things that are affected within depression. We can also see changes in delta phos-B, which are important. And as per that previous slide, when we look at certain models for depression, foot shock, random foot shocks, so the mouse is kind of moving around, it randomly gets a shock, doesn't know when the next shock is coming, so it stops moving. And when it stops moving, we say that it has developed learned helplessness because it doesn't know what's going to happen, so it stops moving. And then we also say that that learned helplessness is a way for us to examine depression, and we can look at the forced swim test as well as the tail suspension test. Is that clear for everyone? Not clear for everyone. Is it clear for some of you? Yeah. Yes, okay, I'll take that as a good sign then. So I told you also that if we took about 60 animals and we tried to induce this learned helplessness, give random foot shocks to these animals, and we took 60 of them, then not all 60 of them will actually stop moving. They won't all develop learned helplessness. They won't all develop depression. Some of them will keep moving. They will not stop. They will continue going even though they're getting these random foot shocks. We say that these are resilient mice. Even though we're using the same strain of mouse, all of them are like C57, BL6J mice, all genetically identical. We still see, even within this genetically identical cohort, some of them don't develop learned helplessness or depression, and we can see there's a correlation between the amount of delta phos-B in certain areas of the brain, outlined for you here, and on the previous slide, it's periaqueductal gray, increases in delta phos-B, or we can also look within the nucleus accumbens, we also see the same thing. Now, we think resilience and depression are opposite sides of the same thing. So if you are not able to get depressed, you are resilient. If you are not resilient, you will get depressed. That may be very, very simplistic, but that's how scientists kind of look at this whole scenario. And you have to take it at face value that this is a correct operational definition. So if you can't invoke if you cannot sort of resolve the stressors that are being applied to you, you do not have resilience, and you're more susceptible to developing depression. So again, in terms of humans, does it correlate? It's hard to say because we have so many other psychological things that work in our benefit. So there are coping strategies that help you deal with prolonged stress. Mice don't necessarily have those, especially lab mice. There is avoidant coping. So if you know that lectures are going to be really stressful for you, you might avoid coming to lecture, which I see many of you have done today. So whatever it is, you have different options, social options that these mice don't have. So the key question is, we have a model that we can induce depression or something that looks like depression. Can we understand the biology? And then can we apply this to humans? Because that's ultimately what we want to do. Is that clear for everyone? That's where we're going? Okay. Anything that I can clear up for you before we move on? So this delta phos-B, as Yi-Wen was asking earlier, not just in the periaqueductal gray, it's actually part of this reward circuit, and it tends to be a transcriptional activator. So we've been talking about VNTRs and transcription factors and turning on genes, and delta phos-B in this context, in this particular series of slides, is an important transcriptional activator. We have a lot of it. It's very important within the brain, and I'm going to show you this on the next slide. It tends to be lower in individuals with stress, and I'm talking here about individuals in an animal model sense. The data in humans is a lot less clear, but it does tend to be very, very strong in animal models with social defeat, learned helplessness, etc. The data on humans, it's not quite as clear. Some people say, yes, we can definitely see lower levels of delta phos-B. Some studies say no change, and some studies say that it might actually be slightly increased. But in animal studies, learned helplessness, social defeat, any of the depression models that you look at, we see a reduction in delta phos-B. So that point is something that you need to know. So are we good with that point? Delta phos-B is a key sort of indicator in animal models that depression is going on. In fact, these animal models can be used to test antidepressant medication. So when you know that an animal is depressed, and if you go back to that original slide or the original video where the rat was sort of struggling, then it stopped struggling, and then it was given an injection of like an antidepressant and it started struggling again. These antidepressants have been shown to reverse the decreases in delta phos-B. Does that mean that delta phos-B is the molecule for depression? What do you think? Probably not. Ooh, downer. Why? Because biology is really complicated. Of course, yes, and it is. It's absolutely true. So we're going to examine that in a moment, but antidepressant medication, those SSRIs, the SNRIs, have actually been shown to increase the production of this transcription factor. So if it goes down during depression, and it's high in resilient individuals that don't get depressed, and antidepressant medications can alter the levels of this transcription factor, we certainly need to consider it as an important player. And again, resilient animals, the ones that don't have depression, seem to have very high levels of this. So important slide, not important slide? Important slide. So, and again, because I was asked to make sure that you are aware of how many questions were coming from each of the lectures, so this would be a second multiple choice test question, and I'm going to tell you exactly how I'm going to ask it. So this is going to be a case scenario. So I'm not going to ask you like, what is delta phos-B? Choose from a list. So if I alter one of the proteins in this pathway, what do you expect to happen? So if I decrease one of these proteins, maybe talking about what should happen, all right? So this slide is important, but I want you to know in advance that I'm going to create a case study or a scenario related to the pathway. So with that in mind, as you are listening to this part of the lecture, if you have any questions or things that are unclear, please ask, because there's likely someone else that is also not clear about this as well. Do you know how to get this onto the big screen, Jeffrey or Stephen? Like, do I just put this up here? Sorry about that. Yeah, oh, well, that was easy. Okay, awesome, thanks. So if we take a look at what we're looking at here, so delta phos-B is the molecule that we're interested in, and delta phos-B might be the molecule that's important in depression, resilience. It might be the molecule that goes up after giving antidepressants. And so what we're looking at here is just a quantification of what I showed you on the previous slides. We're quantitating, we're visualizing these changes that I outlined for you earlier. So remember, we can induce depression in a number of different ways. So in grouped animals, where animals are together, social mice, they're grouped together, they have lots of other friends to kind of play with, we actually don't see any reduction in delta FOSB in the nucleus accumbens. And again, I don't know what your neuroanatomy background is, but we can separate out the nucleus accumbens into two areas based on the types of neurons we find there. And in the outer part of, the inner part of the nucleus accumbens, which is known as the nucleus accumbens core, we see that delta FOSB tends to be pretty high. And in the outside part, the shell, we also see that delta FOSB is high in animals that are grouped together. So they're nice and normal, happy animals. If we have them socially isolated, what we see is there's a dramatic reduction in delta FOSB. So these animals that are socially isolated, where we think that they're going to be depressed, have a reduction in delta FOSB. So do you all see that? Are we good? So let me ask you this question. Would you expect to see the same effect if I gave random foot shocks? What would your prediction be? So would I see the same trend? So is social isolation a model for depression, and is random foot shocks, learned helplessness a model for depression? Yes, so the trend would be the same, correct? Yeah. Okay, so have I lost anyone so far? No, okay. If I lose you, let me know. All right, so if we continue on, and this is sort of just a way to look at a brain section, and we are looking at the relative intensities within the nucleus accumbens in these areas here. The hotter the color is, so red and yellow, it means that we have more of the delta FOSB, and you can see that in cross-section, coronal section, that you see here in grouped animals where they are happy, they are not a model for depression, we see lots and lots of delta FOSB, so in this area. Do you all see that? Yes. Okay, awesome. And if they become isolated, do you see that in the same or representative areas, we see a reduction in delta FOSB? So we don't see as much red, we see very little red or very little yellow compared to the same area here. Yes, hello. Hi.
So just to clarify, are the same mice that are in the group?
No, we're looking at a mouse, and we have sacrificed the mouse. We've cut through the nucleus accumbens and the cortex, and then we've stained it for delta FOSB. And we've taken a different mouse, and we've socially isolated that mouse for a while. We've seen that the tail test suspension is not moving around, it's not swimming, and so then we examine the same area of the brain in the socially isolated mouse, and we see this reduction. Does that answer your question? Anything else I can clarify for you? No? Okay. So if we continue along this line, if that's all clear for you, one of the other tests, so we can look at tail suspension, struggling or not struggling, or we can look at swimming. And the other thing that we can do, if an animal is depressed, especially when they have been socially isolated. So you might wonder, like, what are we looking at? Why have yet another test? If we look at the socially isolated mice, one of the other things that we can see is that if we are looking behaviorally, so we're not looking at the staining intensity like we are at the top, but if we are looking behaviorally in grouped mice, which are nice, normal, happy, not depressed, versus isolated, one of the things that we'll see is that if we now provide a playmate, or if we provide another mouse, and then we see how long they interact with each other, the socially isolated mouse that's depressed, that has a decrease in delta FOSB, just doesn't interact with the other mouse. It just doesn't want to be socially there. Do you all see that? Is that okay for everyone? Is it not okay for anyone? I'll ask it a different way. Okay, so I'll ask it yet another way. So is anyone who doesn't understand it, put your hand down. Okay, awesome. Wait, Jessica, do you not understand it? Okay, never mind. Okay, cool. All right, so it goes down. They spend less time together. So this will be our output. Have we induced depression? Cool? Good? Yes, okay. So one of the other things that we can start to do, now we're looking at these isolated mice. They don't like interacting, so we use this interaction as a way to measure are they depressed or not. So one of the things that we can do is, hey, you know what? Delta phos B is like a really important molecule. It's an important molecule that I tried to demonstrate in panel B that is going down in the nucleus accumbens. In fact, across the entire nucleus accumbens, in the shell and in the core, it goes down. I hope I've convinced you of that. And if I haven't, Eric Nestler probably has convinced you of that. But more importantly, if it's going down and antidepressants make it go up, what if I artificially make depressed mice, mice that have been isolated, what if I introduce delta phos B to them? And so what we're looking at here is the delivery of a virus that will infect all of the neurons within the nucleus accumbens in these isolated mice. And in these isolated mice where we don't see a lot of interactions, can we see any change? And that is these center panels that we are looking at here. Have I lost you so far? Not yet. Okay, so I'm going to start losing you any second now, so just bear with me. So we introduce the HSV virus, the herpes simplex virus. It infects neurons incredibly well, and we are using the herpes simplex virus to deliver GFP. So it's just a marker that, yes, these neurons have been infected, they now glow green with GFP, and does it make any difference to their social interaction? If they've been isolated, no, they still do not interact. Clear on that one? Good? Thank you. Awesome. Thank you. Awesome. Sorry? So the last sentence, in these socially isolated mice that should be depressed, if we inject in HSV that is carrying GFP, the gene, and we introduce GFP, and it has no other effect, it just makes the neurons glow green, it actually doesn't affect their interaction. They still don't interact. Good? Good. Okay. But here's the real kicker. The real kicker is this one here. In this HSV delta phos B, so we use the same viral vector, HSV, and now we are overexpressing, or we are expressing delta phos B. So we've shown at the top in A and B, delta phos B, as you get socially isolated, goes down, 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 down. Now, after it's gone down, they've been socially isolated, they're depressed, they're not interacting, I've shown that. Now, if I inject in, after they have become depressed, after they stopped interacting, can I get them to interact more? Can I reverse depression? Are we okay with that so far? Okay, thank you. So after we introduce this delta phos B and we overexpress it, we actually see a tremendous increase in their interactivity. It goes almost back to the levels that we would expect to see under normal conditions. Can we do the reverse? And this becomes, again, a really cool experiment. So if we try to do the reverse, let's use a different virus. Let's use AAV. And it's another replication deficient virus. These are grouped animals, nice and happy. We expect them to interact really well. And if we introduce GFP, they still interact not quite as well as they did over here, but let's assume that they interact fairly well. But now let's introduce something that disrupts or reduces the level of delta phos B. How do you reduce the levels of delta phos B? Nowadays, in 2019, you might use like a microRNA. You might use RNAi strategies. But another simpler way is to use an antagonist of delta phos B, which is delta Jun D. If we introduce delta Jun D, which binds to and makes sure that delta phos B can't work as a transcriptional activator, we actually see that these grouped animals are actually going to not interact. They are going to look like they're depressed. And we can continue to do this using like Western blots where we can see depressed animals having lower delta phos B. So all of this suggests that if we manipulate the levels of delta phos B, that we can alter the behavior that looks a lot like depression. And again, why is this delta C-Jun D so important? So this is why we're interested in this. So delta phos B, or phos-like protein, and Jun D normally combine together. They form a complex that is called AP1, and this complex AP1 then sits on different target genes in the promoter regions to turn on expression of genes like CREB and others. And so if we have delta C Jun D, it actually blocks delta phos B from working and forming this transcriptional activator. And this is the proposed mechanism by which delta, the bar in red, the delta C Jun D lowers, because it's blocking the delta phos B, it's lowering delta phos B by directly antagonizing it, binding it, so it can't work as a transcriptional activator. So this is a slide that I want you to think about over the next week. And it start with this slide at the beginning of the next lecture, because you are going to see it again on your final exam. As I mentioned to you earlier, from this final exam, on this final exam, 2 questions, multiple choice from today. I would also very strongly suggest for you, as you're making notes today, to also outline all of the different animal models and the way that we look at depression in animal models of depression. In the next lecture, we'll come back and we are going to start to look at the effects of exercise and sleep on these depression models. Have a great week, and I will see you either in office hours or in class next week.
Sure.
Yep.
So my question was actually about the column. So I don't think I made myself clear. So these in statistics, I learned that when you take a larger sample, you get a better estimation of the trend in the population. So wouldn't these larger samples be more valid representations than the
Cascade at all, the output, even in a large sample, because they're also meta-analyses, it depends on their criteria for the meta-analyses.
Yeah.
They assume that it's not as good. So they're careful with the 847 individuals. Their criteria that they use prospectively are different and more precise and more valid than what they're looking at in these larger meta-analyses that show no impact. That's their argument. So in some ways it would invalidate like